In the previous chapter, we introduced the topic of genetics, talking about some of the historical references to Mendel and his work. And now in chapter 13, we'll move on and look at how we came into the modern era of understanding genetics. So remember that Mendel did most of his work in the mid-1800s, working with pea plants, and he published some of his work in a paper in 1865, but it was relatively unknown at that time and not really common knowledge. So his work really wasn't forefront, and we really didn't understand the importance of his work until the 1900s. It was at this time in the early 1900s that four additional workers listed here, each working on their own, came up with very similar material to what Mendel had done, and they rediscovered that Mendel had actually found these results way back in the mid-1800s. Ironically, now one of the researchers, Hugo de Vries, is the one who actually coined the terms gene and mutation. So although we accredit those often to Mendel, it wasn't until this researcher actually came up with the names for those particular aspects of genetics. So looking back at some of Mendel's works, you remember that Mendel came up with a couple laws that he stated, and the laws were based on the statistical results from doing his pea plant studies. But these were purely conceptual, meaning that he derived them purely from the numbers, that he wasn't actually seeing all these answers. And this was mainly because he really didn't know what was passing the information on in cells. He called these things hereditary particles, but he really didn't know that they were pieces of DNA that we now call genes, because those concepts hadn't even been discovered yet. So, because Mendel really didn't know what these things were, what do we need to know in the early 1900s that were able to really show us what these hereditary particles are? Well, first off, we need to have improved microscopes and staining techniques, the use of dyes, so that chromosomes can actually become visible. So here we have a chromosome smear, and these chromosomes are very, very tiny. So we have to use a very high magnification in order to be able to see them in a high quality microscope, which wasn't really possible until the 1900s. Now, today we know what chromosomes are, that they are strings of nucleotide units and along with histone proteins, and that's the material that actually stores the genetic information into those units we call genes. So moving on to the later 1900s, the later 1800s, once we started to understand that chromosomes might be the actual unit of inheritance, we'll take a look at who historically was able to discover that. So if we look at chromosomal theory of inheritance, we know that during meiosis, we've talked about meiosis, those chromosome pairs, they will migrate or move into the gametes as unique structures. So chromosome 1 will stay as chromosome 1, and one copy will go into each gamete. But if an individual has two different copies of that chromosome 1, then when they make gametes, some will have that copy of chromosome 1, some will have another copy of chromosome 1, the other copy. And then there could be a random assortment of the chromosomes 2 and the chromosomes 3, and so forth. So each parent makes gametes that have only half their chromosomal complement. We've talked about that before, that gametes need to have a haploid complement because when we put two gametes together, an egg and a sperm, we need to get back to having two copies of every chromosome. And even though the gametes, the male and female gametes, the sperm versus egg, are very different in their size and the way they look, they still have the same number of chromosomes, meaning that each parent will donate the same genetic information, the same amount of genetic information. And when do these gametes come together? Well, they come together in a process that's called fertilization. So when the two gametes meet, they fuse together, they join, and they each have their own nucleus, their own separate nucleus. The sperm does, the egg does. But then those two nuclei will fuse together and form a single nucleus as the new individual is formed, and that will have the full complement of chromosomes. So as we look, whether it is a fly or whether it's a human, we will see that every species has its own karyotype. So keep in mind that the chromosomal number will vary. So here we see the chromosomal complement in the fly. Here we see it in the human. But we always have two copies of every chromosome. One that comes from the maternal side, one that comes from the paternal side. And the same is going to be true in the fly species as well. Now, the majority of genes are going to be located on what we call the autosomes. Those are the chromosomes which code for the majority of body functions and appearances. But there are some genes which are located on the sex chromosomes. So that is what we call the X and the Y chromosomes. And again, majority of those would be on the X chromosome because the Y is a really tiny little chromosome. So any gene that's found on the X chromosome indicates that that trait could be sex linked, meaning that if the individual is a male or a female may determine what the gene will represent. And so the first demonstration of sex linkage came through using the fly model. And here we see that some flies have red eyes and some flies have white eyes. And that is can be a sex-linked trait. So the research group, Morgan's research group, was the one who actually demonstrated this sex linkage and showed that in the fly, the eye color seemed to be inherited along with the gender of the fly. These did not segregate independently, meaning the sex and the eye color were connected together, and this violates Mendel's second law, which said that traits will segregate independently, that it was not. So Mendel, back in the 1800s, thought that these genes didn't interact with one another at all and they weren't tied to one another. But Morgan's work showed that, in fact, they could. So this proved Mendel's second law not to always be the case, that sometimes genes will be expressed together and that those traits are tied to one another, meaning that they're on the same chromosome. They found that when other traits were examined, that those that happened to be on the same chromosome did not always segregate independently. So that meant that they would in fact be tied to one another. Traits on the same chromosome did segregate, however. So that meant that if you are on the same chromosome, then most likely you're going to be together. But if the traits happen to be far apart, meaning like if you have one trait way up on the top part of the chromosome and one way on the bottom part of the chromosome, then those traits may end up being not being tied to one another. And so the question for that is, how and why could that occur? The answer is the process of crossing over. So if you remember back to meiosis, in meiosis 1, there was an opportunity where homologous chromosomes could do a little swapping of parts. It's usually the very ends of the chromosome, so that if a part got swapped with another chromosome, then it wouldn't always be tied with the genes that were previously on that same chromosome. And this allows for variability. So in this diagram, we're going to take a look at some genes that are linked and some genes that are not linked. So we assume that if a gene happens to be on the same chromosome, then it's going to follow through and always go to the gamete being the same. So this would be the case that we see in B when we're looking at flies that have gray bodies. A capital G represents a gray body. A small g would represent a black body. A capital L means a long wing, and a lowercase l means a short wing. So when we look at genes L and G, they happen to be on the same chromosome, but they're fairly close together. Then we're not going to have any crossing over happening. And if we have no crossing over happening and we have this individual who is heterozygous, meaning two copies of each of the genes, a big G and a little g, a big L and a little l, they happen to both all be part of this one organism. This organism is going to be able to produce only two types of gametes. It's going to be able to produce only a chromosome that has big G, big L, and again, or little g, little l. So that's the only kind of gametes that this parent can produce. If we cross this parent with a second parent that has only little g, little l, i.e. homozygous for little g, little l, then you see the possible offspring, but we can't ever get this kind. We can't ever get a big G, little g, two little l's, because that is just not going to be a possibility for that organism. And so we get a 50-50 representation of these. But if we do have two genes, so this is C, two genes L and G on the same chromosome but far apart, this time they're far apart, then we can have crossing over happening where we're going to swap some of these L's, big L's and little l's, and now we can make all four different kinds of gametes for this parent, and we cross it with the same fly, we can end up with representation of all of these. And when Morgan did that, that's what he found out. He did find that he got all four types, indicating that those particular genes must be on the same chromosome, but fairly far apart, so that you do in fact get some crossing over happening. So when that crossing over happens, here's another representation. So originally you have a blue chromosome here, that's a parent, and it's capital A, capital B, and you have the second parent one, which is the green chromosome, little a, little b. Then you make a copy of each of these. So you're going to make another total green line. You're going to make another total blue line when you're going to make gametes. But instead of having two complete blues, two complete greens, we're going to allow crossing over to happen in meiosis 1, so that a bit of the green which carries the small b, is now going to be part of a capital A chromosome. And a part of the capital B chromosome is going to now be part of the small a. And so what we end up is with these 4 different possible chromosomes that we can put into gametes for this organism, and instead of having just 2 possibles. And these middle ones, because they have been recombined, we've combined from 2 different chromosomes, they're called recombinant chromosomes. So we have the ones that were original in the parents, and then we have 2 new ones that are going to be possible to get passed on to the gametes. Again, all of this increases variability. Further evidence for this came along by a student, Alfred Sturtevant, who was working in Morgan's lab and researching with these flies and looking at these positions of chromosomes, genes on the chromosomes. And he was actually able to set up a mapping, a way to represent distance on a chromosome. So he came up with these units of distance. And so here, this would be where the gene is for this trait. Here was that gene for the body color. Here's one for eye color. Here's one for wings. And there's multiple genes for eye color. So here's another gene for eye color. And so these are distances down the chromosome. You can see that these 2 are close or closer. These 2 are even closer to one another. And then you have increasing distances. This one and this one are fairly far apart. So he found that we could look at a recombination frequency was a function of the distance between the genes. The likelihood that there could be recombinance, that we could have a crossing over event, was dependent on how far apart the genes are on the chromosome. When we take a look at a human karyotype, this is one for a female, a human female, in which we are showing, don't worry about the coloring, that's just indicating banding patterns. And so that gives you a good idea that they're pretty similar between the 2 types. But we have 1 through 22 pairs of what we call autosomes. So these are the everyday body chromosomes that are coding for all kinds of things that our bodies need to do and to show, whereas the last set are called our sex chromosomes. And while these are very similar to one another as far as positioning, the pair in the sex chromosome group, the X and the Y, are extremely different from one another. They don't have the same genes on them. They're not even the same size. So what was found was that to be a female, you would have 2 X chromosomes to be a genetic female, and to be a genetic male, you would have an X and a Y chromosome pair instead of the XX. Now, what can happen if during the production of gametes, chromosomes don't break apart from one another and allow for only 1 copy of each chromosome to go into each gamete? When that happens, it's called non-disjunction. So disjunction would mean a separation. Non-disjunction is kind of a double negative. We're not allowing the chromosomes to separate. So remember, we have a meiosis 1 and then we have a meiosis 2. During meiosis 1, the 2 pairs of chromosomes, so say this is chromosome 1, two copies of it, and then there's a copy of a copy. So there's 4 total chromosome number 1s, and say this is chromosome number 2, 4 copies. During meiosis 1, we're just going to split up the 2 pairs. And then in chromosome 2, we will actually split apart the pairs. So if we have non-disjunction in meiosis 1, that means this is what would normally happen. We split apart the 2 pairs, And then here we don't. Both pairs go to one side. When that happens, all of this goes to one cell and only this goes to this other cell. So when we go through meiosis 2, the chromosome number 2 will separate just fine, and you notice here we have only one copy of each of those. But for chromosome number 1, we're going to have two copies, the blue and the red. So that is too many chromosomes in these cells, and that can result in an abnormal individual when this gamete becomes fertilized. Any of these. So these have an extra chromosome. These are missing a chromosome. The same kind of thing can happen in non-disjunction can happen in meiosis 2. So this is when it happens in 1. This is when it happens in 2. So at the end of 2, we have a normal splitting of a copy of chromosome 1 pair and chromosome 2 pair, and chromosome 1 pair and chromosome 2 pair. Here we have normal division. Remember, the pairs come apart, and so we have one copy of chromosome 2, one copy of chromosome 1, or the other way around. But in this one, we noticed that both of these stuck together and didn't separate, and they got pulled to one side. So here we end up with 3 copies of chromosomes. We should only have 2 individual chromosomes. We have 3, and here we only have a single one, so we're missing a chromosome. So the individuals that would be fertilized, say these were eggs, if they were fertilized with a normal sperm, are still not going to be normal because they were given the wrong number of chromosomes during meiosis at some point. Now, ploidy, the word ploidy has to do with the number of chromosomes. The number, just the number. Doesn't say you have too many or too few. If we put the prefix on here, euploidy, eu means true. So this means what we expect to find. So euploid means the expected number of chromosomes. In the human, that would be 22 pairs of autosomes, 1 pair of sex chromosomes, for a total of 23 pairs, or 46. An aneuploid means you have a wrong number of chromosomes. So something has happened to an individual chromosome. It has one extra chromosome, or maybe too few. And here's a couple examples. Here's an example of monosomy, where we're missing a chromosome. That happens in a condition called Turner syndrome, where one of the X chromosomes doesn't properly separate during non-disjunction of the egg, and you end up with only one X chromosome. So you probably get no X chromosome from the egg and one from a sperm. So you have an individual that only has one sex chromosome. This O is not an O, it's really a 0. Trisomy, tri means 3, means an individual went through non-disjunction and has ended up with 3 copies of a particular chromosome. In this case, it's chromosome 21. That's what we call Down syndrome individuals that have inherited 3 copies of chromosome 21 instead of just 2. And then polyploidy. Now this is different from aneuploidy. Polyploidy means you have a whole extra set of chromosomes. So, for us, that would mean you would have an extra set. So not 23 plus 23, but 23 plus 23 plus 23. So you would have a whole extra set. Now, this is very rare in animals. Does not really happen much in animals, but is really common in plants. It's common in plants to have maybe up to 4 copies of their haploid genome. Here's a visualization of that splitting up and the non-disjunction possibility. And then I want to talk to you about an interesting case which can show non-disjunction of the sex chromosome, and we're going to see this in cats. So this can happen normally in animals if we're talking about the X chromosome. Because what happens normally in females is that if females kept both of their X chromosomes, then they would have extra genetic material than the males because the males only have one X chromosome. So what normally happens in most of the body cells of a female is that randomly one of the X's is inactivated. It kind of just shuts down. It's not that it goes away, but it's just not functioning. And so that's called X inactivation. Now in cats, the color, the gene for their fur color is actually on the X chromosome. So it's on the X chromosome. For females, some of those X's are going to get turned off. So that's what I'm mentioning here. 2 X chromosomes in the cell, one's going to get inactivated, meaning you could have a cell here that says, okay, I'm going to keep on my black fur color gene, but I'm going to shut off the brown one. And this cell over here might say, I'm going to turn off the black fur chromosome, but turn on the brown one. And so that way, if a cat has 2 different alleles for coat color, if it has 2 alleles for black, then every cell is going to have black. But if it has one allele, one X that codes for black and one that codes for brown, then there could be a random splotching of black and brown throughout the coat. And so this last question, why does this happen in females but not males? Well, of course, in males, they only have one X chromosome, so they won't be deactivating any of that. So you won't find this kind of coloration in a male cat. So anytime you see a tortoiseshell cat, which is what this is called, it's not a tiger cat, it's a tortoiseshell cat, then it is a female and always will be a female. Okay, so that will complete this chapter.